Hello students, welcome to EPG Pathshala. I am Dr. Kajal Jindal from University of Delhi. Today we are going to discuss about the module Fluorescence Microscopy from the paper Characterization of Materials 1. So students, let us see what we are going to learn in this module. In this module, principle of fluorescence microscope is described. Epifluorescence microscopy is discussed in detail. In this module, the various types of immunofluorescence are studied. Also, the instrumentation and working of total internal reflection fluorescence mode of fluorescence light microscopy is discussed in detail. At the last, we will study the advantages, limitations, and applications of fluorescence. The formation of an image using fluorescence is referred to as fluorescence microscopy. The setup could be simple, resembling an epifluorescence microscope, or complex like confocal microscope that uses optical sectioning to obtain superior resolution of fluorescent image. Eric Petzig, William Marner, and Stephen Hill developed super-resolved fluorescence microscopy, bringing the optical microscopy into nano dimensions. Fluorescence microscopes create enlarged pictures of fluorescent light emitted from excited molecules of a sample by employing special filters and specific technique of illumination. Filters that are used in the microscopes are configured such that they isolate and control two separate sets of excitation and fluorescence wavelengths. One set which contains the shorter excitation wavelengths from illuminator, and the filters is directed towards the sample, and the second set which contains the longer fluorescence wavelengths coming from the sample creates a picture of the sample in image plane. Proper selection of fluorochrome, filters, and illuminator is imperative for efficient working of fluorescence microscopes for the particular applications and for determining the quality of fluorescence signals. Let us discuss the principle of fluorescence microscopy. Figure 1 illustrates the schematics of the fluorescence microscopy technique using the most popular Zablonsky diagram, wherein the electrons are excited to a higher energy level in fluorochrome by the absorbed light. The electrons lose some energy by vibration and rotation, while the remaining energy is released in the form of fluorescent light when these electrons relax to their original energy level. This happens approximately 10 nanoseconds. Wavelength light. The fluorophores present in the sample absorb the light and then emit a longer wavelength light. Spectral emission filter separates the illuminated signal from weak emitted fluorescence. The elements of a fluorescence microscope include light source, excitation and emission filters, and dichroic mirror or beam splitter. The filters and dichroic beam splitter are selected to match spectral excitation and emission characteristics of the fluorophores are employed to mark the sample. The distribution of an individual fluorophore or color is pictured at a time. Several single-colored images can be merged to obtain multicolor images of different fluorophores. Excitation of the fluorophore and the subsequent visualization of fluorescence are performed by the objective of epifluorescence microscopes. Advanced microscope designs include confocal microscope and total internal reflection fluorescence microscope, which is also known abbreviated as GIRF. Let us now discuss the epifluorescence microscopy in detail. Objective lens focuses the excitation light on the sample, and the fluorescent emission from the sample is directed to the detector by the same objective lens. Lenses having high numerical aperture offer better resolution. Only reflected excitatory light reaches the objective, which is then transmitted through the specimen with emitted light, resulting in high signal to noise ratio. This is achievable by epifluorescence method. As can be seen in the figure, the dichroic beam splitter acts as a wavelength specific filter, and it transmits only the fluorescent light towards the eyepiece through detector and reflects back the remaining excitation light to the source. Let us now discuss about the light sources. Different light sources which are employed include xenon arc or mercury vapor lamps along with the excitation filter, lasers, supercontinuum sources, and high-powered LEDs. By placing two microlens arrays in illumination path of a wide field epifluorescence microscope, extremely homogeneous illumination having a variation of 1 to 2% is attained. Now, the sample preparation. For the sample preparation, the fluorescent samples must be used. There are various methods to prepare fluorescent samples, such as the fluorescent staining, labeling, the expression of fluorescent protein. Then, using intrinsic fluorescence of the specimen, which is also called as autofluorescence. This can be achieved by various techniques which are used for fluorescent staining. Let us now discuss about immunofluorescence. The technique of immunofluorescence employs highly specific binding of an antibody to its antigen and then labeling the specific proteins or other molecules in a cell. Initially, the specimen is treated with the molecule specific primary antibody. A fluorophore is then combined to this primary antibody. Otherwise, a secondary antibody attached to a fluorophore can also be used to specifically bind to the first antibody. The following types of immunofluorescence are there. First one is the direct immunofluorescence. Second is the indirect immunofluorescence. And third is the microimmunofluorescence. Let us discuss about the direct immunofluorescence. Direct immunofluorescence is employed for detecting antigens in clinical samples by using antibodies which are labeled with specific fluorochromes. The process involves following steps. Firstly, fixing the smear onto the slide. Then, the treatment with labeled antibody, followed by the incubation, then the washing to eliminate extra unbound marked antibodies, and finally the observation using fluorescent microscope. When observed under fluorescent microscope, the field is dark and areas with antibody binding exhibit green fluorescence. The figure also shows the direct immune fluorescence having a linear IgG band at the basement membrane in bullous pemphigoid. We will now take up the indirect immunofluorescence. This technique is different than the direct immunofluorescence in that it detects the antibodies which are present in the patient's serum. This antibody is then raised in mouse and antigens on smear are allowed to react with these specific unlabeled antibodies. The unbound antibodies are then washed off. Another antibody is added to detect the presence of specific mouse antibody which is bound to the antigen on smear. The second antibody is labeled anti-gamma globulin, that is, rabbit antibody against mouse antibody. This binds to the Fc section of first antibody and remains even after washing. The presence of the second antibody can be ascertained by viewing under fluorescent microscope. Autoantibodies can be detected via this technique. Additionally, this technique is widely employed for detecting anti-nuclear antibodies, that is ANA, which are observed in SLE patients' serum. The figure shows the indirect immunofluorescence, which is showing intercellular deposits of IgG in pemphigus vulgaris. We will next discuss about microimmunofluorescence. It is a serological technique which is used in detection of antibodies in patient serum. Its working principle is analogous to that indirect immunofluorescence. However, this technique is done on Teflon slides having several wells dotted with antigens. Microimmunofluorescence is employed in the serodiagnosis of Q fever, Mediterranean spotted fever, detection of IgG, IgA, and IgM antibodies, chlamydia, toxoplasmosis, epidemic typhus, etc. of many intracellular events such as endocytosis, exocytosis, cytoskeletal, and dynamic single molecule imaging. TIRF is one of the latest developments fluorescence microscopy of biological systems, and it provides ultra-high axial resolution on the scale of 100 nanometers at the sample or cover slip interface. Such high optical sectioning resolution at the sample or cover slip interface leads to very high signal to noise ratio for studies including direct observation and recording of cell membrane function, vesicle fusion, and membrane proteins, etc. This microscopy technique is extensively used in the experiments involving low signal intensity and fast imaging. These standards are achieved via electron multiplying CCD, that is EMCCD technology, which is being studied by researchers on an intensive scale. Let us discuss the various steps which are involved in the working of a typical TIRF. The steps involved in a typical TIRF experiment are as follows. Firstly, two laser beams are adjusted to the microscope port height by employing periscopes. These laser beams are then combined via a dichroic mirror which is located on a kinematic count. A set of two erasers are placed at same height along the optical table such that the beams traverse the same optical path. The illumination spot is enlarged to about 60 micrometer in diameter by using a 5x beam expander on the XY translation stage along the optical path. However, a combination of 10x beam expander and tunable iris set at a diameter opening of 1 centimeter may be employed to achieve equal illumination area for both lasers if the two laser beams have substantially different beam diameters. The chromatic aberrations present in the beam expander can be minimized by using a set of achromatic lenses. The beam is then directed towards a kinematic mirror kept on a linear translation stage. This mirror reflects the light at 90 degrees and sends towards the back illumination port in case of an inverted microscope. This step is optional. A suitable filter set is placed in the filter turret of a microscope and a circular field diaphragm is placed on the nosepiece. The mirror position is adjusted such that the beam center coincides with the center of object's plane. Along the optical path, an achromatic doublet TIR lens is placed with an XYZ translation stage. The illumination area size is determined by the focal length of the achromatic doublet. Achromatic doublet TIR lens is typically between 25 to 50 centimeters. Firstly, the lens position is adjusted such that the beam passes through the center of the lens. And secondly, the Z position is adjusted to focus the beam onto the center of nosepiece of microscope. We will now study the applications of fluorescence microscopy. The intracellular macromolecules distribution in various structures. Example, membranes, cytoskeletal filaments, and chromatin can be analyzed using fluorescence microscopy. This technique can be employed for studying the mechanisms of binding dissociation as well as diffusion, that is, fluorescence recovery after photobleaching, which is abbreviated as FRAP, associated intracellular dynamics in macromolecules. Nearest neighbors of proteins, interaction states, as well as reaction mechanisms can be studied via fluorescence energy transfer (FRET) and fluorescence correlation microscopic technique. In vivo studying the dynamics of single tagged molecules or assemblies of molecules by employing the superior sensitivity technique, which is termed as total internal reflection fluorescence, that is TIRF microscopic technique. Also, intracellular ionic concentrations as well as changes in concentrations of various other ions such as H plus, Na plus, K plus, Cl minus, etc., and some metals, etc., can be determined. Then, the experiments involving specific labeling of organelles and cytoskeletal proteins using dyes can be studied. Moreover, rate and extent of enzyme reactions can be determined by using conjugates of fluorochromes whose fluorescence is changed by activities of enzymes. Is readjusted such that the laser beam is projected on the ceiling and the Z position is adjusted to achieve minimum achievable diameter of laser beam. Majority of the objectives suffer from chromatic aberrations, which make it impossible to simultaneously focus multiple laser beams. Thus, either the Z position of the TIR lens is tuned or an intermediate position should be found before switching between different laser beams. Objective: a flow chamber with surface immobilized fluorescent microspheres on a covered slips prepared and placed onto the microscope stage. The objective lens is focused onto the sample by using binoculars to identify the immobilized beads on the cover slip surface. Images are acquired with the charge coupled device camera, which is located on a plane that is conjugate to the object plane. Adjustments of either X or Y position of the TIR lens leads to a shift in illumination area which is away from the center of the CCD chip. The X or Y position of 5x beam expander or the mirror when used, and then the TIR lens needs to be readjusted to match the center of the illumination area well with that of the CCD chip. Let us now discuss the advantages and limitations of fluorescence. Let us first the advantages. The fluorescent microscopy technique offers numerous advantages. The first one is that signal to noise ratio is very high. That is because of the black background. Then there is a linear correlation between the fluorophore molecules and output signal. Then there are a large number of fluorophores which can be used. Additional parameters such as FLIM and FRET are present. Specimen can be examined by fluorescence microscopy. Photobleaching can be avoided by employing more robust fluorophores, reducing the illumination, or applying photoprotective scavenger chemicals, etc. Though the fluorescence microscopy, by using fluorescent reporter proteins, has facilitated the study of living cells possible, cells are prone to phototoxicity, especially when illuminated by short wavelengths. Under illumination, fluorescent molecules tend to produce reactive chemical species, thereby enhancing the phototoxic effects. In contrast to the transmitted and reflected light microscopic techniques, only the structures which are marked for fluorescence are observable in fluorescence microscopy. So students, let us summarize what we have learned in this module. In this module, the principle of fluorescence microscope was described. Epifluorescence microscopy was discussed in detail. Then, then the various types of immunofluorescence were studied. Instrumentation and working of total internal reflection fluorescence mode, that is TIRF, of fluorescence light microscopy was discussed in details. Finally, the advantages, limitations, and applications of fluorescence were discussed. Thank you, students, for your attention.